So, okay, we've just obviously went through Meiosis 1 very quickly, just to do a bit of a revision. And what we worked out was that at the end of Meiosis 1, so once you had your telophase 1 and cytokinesis 1 happening.
Sorry.
Can I speak?
Yeah.
Okay, cheers. Once you had your telophase 1 and cytokinesis 1 completed, you ended up with 2 cells, and these 2 cells contain half the number of chromosomes that the original cell contained. So remember, I was drawing original cell with 4 chromosomes, so then each of the resulting cells contain 2. In reality, you would go from 46 chromosomes all the way down to 23. So then the question is, well, why do we need second division of meiosis at all? Because seemingly we achieved what we set out to achieve. We achieved the fact that you have half a number of chromosomes, and also we achieved the fact that these chromosomes are different to the original ones. And the reason why they're different is because you had your crossing over, where you swapped segments of chromosomes. And you also remember we had another mechanism called independent assortment of homologous chromosomes. Do you remember that one? Where we had to calculate how many options you can have by switching chromosomes across the equator in metaphase 1. So why do we need that meiosis 2? Anybody's got a suggestion? What's wrong with these cells?
You've
Why
still
can't we
got
just
the
store?
sister chromatids are still connected.
Right, absolutely right. So each chromosome here, even though you halve the number, but each chromosome still contains 2 sister chromatids. And what I need to achieve in reality, I need to go back to each chromosome just containing one local DNA, one chromatid. So at the moment, you can see these are your sister chromatids. And what I need to do is separate those out and put them into separate cells. So hence why you need to go then into the next division, which is your meiosis 2. Now another thing which is worth noting is that you remember you had interphase before meiosis 1, but you do not have interface between those 2 divisions. So you do not have interface between meiosis 1 and meiosis 2. So I'll just write that down quickly. So no interface. So you go from telophase and cytokinesis of meiosis 1 straight into meiosis 2. So again, another reminder, traditional to use Roman numerals here. If you're unsure how to type them in your assignment, well, Roman numeral 1, that's just capital I, right? So then 2 is just 2 capital I's side by side. Okay, because sometimes people start searching for symbols or something complicated, actually it isn't. That division then, meiosis 2, actually it's much more simple compared to meiosis 1, because meiosis 2 is very similar to mitosis. And hopefully, because we've done now mitosis a few times and we did a few rounds of revision, you should be a lot more comfortable with that. Now, what I'm going to do is, I'm going to pick this cell, right, which has got red and blue chromosome, or you can use 2 of your favorite colors. And I'm going to show you what's going to happen to this cell in meiosis 2. And keep in mind that exactly the same thing is going to be happening to the second cell. So both of them are going to undergo the same stages simultaneously at the same time. But I'm not going to draw 2 lots, I'm just going to draw it as using that one cell as an example. But as I said, the same thing is going to be happening to the other cell. So that's my cell. So, as you should be able to remember, the first stage of division is prophase. So in this case it's prophase 2 because it's prophase which belongs to the second division. So what would you normally associate with prophase? You should know it now by heart without even looking in your notes. So what would normally? Spindle forms.
Membrane starts to disappear.
Right, which membrane? Let's be more specific.
Cell membrane.
Cell membrane starts to disappear.
Nuclear.
Nuclear membrane. Can you see the difference? Cell membrane is that line, nuclear membrane is that line. If I dissolve cell membrane, the whole cell will disappear. So what I'm dissolving is nuclear membrane. And that's pretty much it. Do I need to condense chromosomes? Maybe slightly if we decondense a little bit at the end of meiosis 1. But for most part, majority of cells don't actually bother, if you like, to decondense chromosomes. They just stay as chromosomes between those, because remember there's no interface here. So for most part, you probably don't even need to condense chromosomes, or if they did decondense, you just sort of go back to them being fully condensed. So my picture would look a little bit like that. So again, depending whether it's early or late prophase. So you'd have beginning of your spindle fiber, the nuclear membrane will going to be starting to disappear, and your chromosomes would get attached again to those spindle fibers. So something like that. So that would be picture for late prophase, and a picture for early prophase would be something like nuclear membrane just disappearing and spindle is starting to form somewhere on one side. So still have chromosomes inside. Okay. So next one, the metaphase 2. So what would be normally happening in metaphase? Or what would you expect to be happening here?
Where they align.
When they say they, what do they mean by
Chromosome.
they? Okay. Now, can you all have a go yourselves at drawing what you would expect this diagram to look like? And then I'll go and have a quick look. So before I draw anything, can you have a go? Sorry to go again. Have it as a nuclear membrane by mistake. Okay. Do you have some colors that you use, yeah? Because I think it's easier to make sense if you use different colors for different things. Yeah, I mean that's fine, but it needs to be right in the middle though, yeah, so I think that's sort of important. Because the dotted line is the middle, then you can kind of shift things slightly. Yeah, but correct, that's it, okay.
Want to have a quick look? Yeah.
Right.
Cool. Yeah. How's your H?
Okay. It just paints it all in 5 minutes. Right, now, there's going to be a bit of a problem if you draw them like this. So if you draw them like this, what you are running now, where is your equator? Which way is the equator going to be?
I don't know, just put them in a line.
But which line is the question, isn't it? Because if you have line, is line going like this?
Well, like that or like that, but
Well,
if
yeah,
it's
but
like that,
which-
then it needs to be on top of each other, yeah.
Yeah, so can you redraw it then? Because if your spindle gathers here, and you remember, they're going to get drawn apart. If you were drawn it, then that one would be drawn apart. Can you see what the difference is?
Yeah.
Because you would either have to redraw the spindle facing that way, or you have to redraw your line facing that way. They can't be both positionally. Okay, good. Right. So I think most of you got
Maybe that's not really hard.
Thank you.
I think I've got an A4 as well. About what it looked like in the
So I'll just sort of free up a little bit of space. So what you should have drawn is your spindle fibers. That's your middle line. And then the chromosome should be positioned on this middle line, one underneath the other. So one of the common mistakes, not saying any names here, right, is if you draw your spindle like this,
So I did before.
yeah? And this is I can understand where this mistake is coming from. I totally get it, right? So then you've got one chromosome here, and you're lining it up like this. Now, the problem with that picture is that what you need to line up is individual chromosomes, single chromosomes, whereas that picture would suggest that you're lining up pairs of chromosomes, which was what was happening in metaphase 1, but that's not what's happening in metaphase 2. So just be careful how you draw that picture, how you interpret it. Because if we all lined like this, the red chromosome would fully go to one side, the blue one would go to the other side, and that's not what we want. So it's not that picture, it's this one. Right, so next stage, let me have a little bit of space here, anaphase. Anaphase 2. Right, what would you normally expect to be happening in anaphase 2? And this is, you remember, it's very similar to mitosis.
You start moving towards the pot.
Okay, so when you say they, what is starting to move?
The chromatids.
Okay,
It's
not
not
Yeah, so
separated.
yeah, so when sister chromatids separate, yeah, and once they're separate, what do we call them?
Chromatids.
Yeah, well, specifically,
Oh, daughter.
daughter chromatids or daughter chromosomes.
Chromatids.
A bit of a disagreement there going on. So do we agree that sister chromatids separate?
So we've gone from sister chromatids to daughter chromosomes.
Yeah.
Yes, correct. So sister chromatids
I-
separate.
I hate it. I hate it. Even this thing I printed out wouldn't say that.
So sister chromatids separate, and then at that point they are known as daughter chromosomes. They become daughter chromosomes. It's a lot of CH words, isn't it? Right, so same task as before. Chromatids, sister chromatids separate, and then they become then known as daughter chromosomes. Right, so same task as before. Can you have a go at drawing me a picture of what you would expect anaphase look like? And can you pay attention to how you are positioning these chromosomes? Are you drawing them like this? You know, remember those flying birds? Or like that, you know? This way or that way? And then which side? So try to remember what we were talking about, how the chromosomes getting dragged through cytoplasm, and you're holding on to that centromere, which is the middle bit. So think about, as you're dragging them, which way would the ends of the chromosomes invert. Alright, so again, I'm going to give you a couple of minutes to figure it out.
The dragon one.
So you see, that's the problem, if you just use a pre-printed picture, when it doesn't, you're not really thinking for yourself. So that's why I sort of, I would prefer you to draw rather than rely on existing diagrams.
Chromosomes.
Okay. What's that in there?
Can't think of
Okay.
that.
Just want to draw it anyway.
That's sister chromatid, yeah.
Right, anybody finished the picture? Yeah, okay, good. I'm not sure why, you see, they can't be on the same spindle either. Oh, hold on. You're not thinking. Think about what's going to happen. So you're going from this picture, can you see that red and that red, we're going to separate group. Then when
Right.
so there's no way you have two blue bits on one side and two red bits on the other side.
The chroma
Then
would
you
split,
see,
so
you
your
can't
half
go
goes
from
up
that
and
picture to that picture. You have to redraw
That's
that.
Is
the
it drawn the
way
same,
So
the same
just,
way?
just one. So like that.
Is it drawn the same?
Well, think about this, you're
That's so
dragging
neat.
them. Watch which bit, try to visualize.
I want to rip this paper off, but it's got the right side.
They did it on that side.
I've got something to
So
say.
you draw it, right?
So it's like that.
Right.
Like a bird.
Birds wings to be flying that way.
Thank you.
Yeah, you see those as very short. I mean, it captures the idea of separation, but it doesn't necessarily show you how big it is in drawing.
So it needs to be the other way around.
Well, it needs to be necessarily have to wear. If you want to be, it should have been like more like that. You see what I mean? Because you're holding on that middle bit and you're dragging it in that direction, so those bits are going to get
Go in that
go
direction.
in that direction. But you just have to sort of try to picture it in your mind's eye. Sometimes people, but yeah, the idea is correct. Okay, we'll just have a quick look at others.
Then is that one the same way?
Yeah.
That's it.
So that would be So that would be
The only thing is, no, the right idea, the only thing, if you're trying to show them that kind of thing, don't forget, they're going to be the vision on this side, they're going to be pushing
over
right
there,
there, and not
yeah.
pointing over there.
Yeah.
You have to picture it, because you're holding on to that middle bit and you're dragging it, so those bits are going to be pointing that way, you see what I mean?
So that will be that way.
So those ones should have been on this side, and these two should have been on that side. Yes. Yes, correct.
Yeah.
Everything is the colors, yeah. Okay.
11:11.
So I think most of us go there in the end. So let me just quickly
Somewhere different, somewhere.
sketch.
There's a countdown. The countdown starts like that. You guys are being sick of it. Prepare to be sick of it.
Right, so as we were talking about sister chromatids when separating, so I need two red bits. So one going like this, and one going like that. So these are my two, what used to be sister chromatids, now separated, yeah? And as you're dragging them, the ends of chromosomes could be start pointing towards that midpoint because you're dragging it through cytoplasm, so there's a bit of resistance there. That's why they look not straight, but sort of angular. And then the same thing is going to be happening in
That's
the blue.
I've got the direction.
So something like that. So then these are your daughter chromosomes.
She messaged me. She has. In bed.
Right, and then lastly, telophase and cytokinesis. Okay, so same thing as before, same little task. So can you draw me a diagram which would represent the end of telophase and the beginning of cytokinesis, or cytokinesis progressing? So again, think about how we drew those pictures for mitosis stage and just apply the same logic. Telophase and cytokinesis too.
can turn it drawing.
That's it.
I think it's like those the same on there. Yeah. It's cytokinesis is basically when the thing is being
This is
separated.
when you share when it's finished.
The top is when it's pinched in it and then a cytokinesis when it's actually separated.
No, no. Telophase
No,
is
that
about
year was like,
the chromosomes
Yeah.
getting to the end.
it's just, I'm just thinking of you need to worry about shape, but once you're in that, you don't need to worry about the shape.
Cytokinesis is that division of cytoplasm. That's where
Yeah.
that's where the pinch reaction is. Yeah.
So once it's
So you go, yeah. And at that point, you see that they're no longer pointing anyway. They're going to straighten out again. Yeah. So at that point, they'll just straighten out.
Yeah, okay. I like the first stage, it's easy to read. I'm not going to create some...
Yeah.
Oh, let's do both big panel.
Are you doing it? Oh, it's up to you. I usually combine it into one because telophase rarely happens without cytokinesis. That's all. So for the most part.
So is this okay, yeah?
Yeah, yeah. Right, okay. So just to remind you and remind myself, so what are we expecting to be happening in telophase? Hmm? Spindle disappears. What else is going to be happening in telophase normally?
The membrane starts to
Right,
form.
which membrane? Nuclear membrane going to start
Both of them,
reformed.
so
Exactly, so
two.
you'll have two of them. And cytokinesis
The division through
is going to be formed. This is like that pinching action. So one of the common then pictures would be something like that. So my nuclear membrane getting reformed. What's going to be happening also to the chromosomes at that point? They straighten out and they decondense, means they're going to go longer and thinner. Now, depending which stage of telophase, whether you're drawn early telophase or late telophase, I'm going to go for late telophase if you don't mind. So I'm going to show my chromosomes if they're already gone long and stringy, but leave them as we've drawn it, if you've drawn it like little, you know, thick kind of sausages, packages. That's fine as well. So if you don't mind, can I wipe some of this off now, right, just to free myself up a bit of space.
it's two.
So then, at the end of the whole process, right, if I just now go back to that picture,
Yeah, 1, 2, 4.
so then at the end of telophase, what you're going to have is the complete separation of cytoplasm.
Yeah. It should only be half of each, yes,
Yeah.
to daughter, which then will form in. Yeah. So then
So then you end up with, on my picture, 2 chromosomes in each of those resulting cells. But you remember that as well as that cell, the other cell was also undergoing meiosis 2, even though I didn't draw every stage of that process. But by the end of
I've just drawn.
meiosis,
Isn't it?
1 up there.
with that cell, you'll have very similar picture, right? So you'll have 1 black chromosome, if you follow my colors, and 1
We
blue
like
one,
worms, ew.
right?
That's the end of it, isn't it?
Does that make sense, right? So these 2 cells originated from that cell, and then these 2 originated from my green and black cell. Right? So these are your 4 resulting haploid cells. Remember, haploid just means that they contain half the amount of information. And each of these cells could Yeah, maybe, no, that's the end of it, it's not going to split any further. We ended up with 4 resulting cells. And but each of these cells could, for example, become sperm cell, right? If they're sitting in male testes, for example. Yes?
Yeah.
Good. All right, lovely. So, Victor, do you mind switching off the recording?